My name is Sophia Rubsamen, and today I will be discussing a research article titled A Bioinspired Analogous Nerve Towards Artificial Intelligence. This study focused on advancing artificial neural systems. Artificial neural systems are types of computing systems designed to mimic the way the human brain analyzes and processes information. They can restore touch and other senses to disabled persons, cause muscles to activate, and can even provide interactive feedback of robotic manipulation. Advancements in these systems include features like a bionic sensor, which can detect a stimulus, a signal cable, which can mimic axon function, and synaptic transistors, which can detect the location of a stimulus in time and space. While these advancements may sound very fancy, they are also very complex, with very intricate structures, which make them difficult to easily produce. They also face issues in transmission of signals due to interference and consume excess energy than would a typical neural system. This study aimed to develop an artificial nerve that would address these issues and mimic the functions of a biological nerve as accurately as possible. The researchers proposed an artificial perception and transmission nerve, APT nerve for short. This is an all-in-one bionic sensory transmission nerve that can perceive, recognize, and transmit a mechanical stimulation. To understand the research, it's important to understand the basic components of a biological nerve which must be replicated. A biological human nerve detects mechanical stimulation via stimulation of a mechanoreceptor. It also consumes low or no energy without the stimulation because it won't be active in the absence of a stimulus. A biological nerve transmits the information received from the stimulus through connections between neurons called synapses, and it can recognize the location of the stimulus relative to the location of the neuron which it activates. Mechanosensation is the way in which mechanical stimulation is translated into neural impulses. Mechanical stimulation itself is basically any sort of stimulation that applies pressure and creates tension, which elicits a reaction. This is the basis for experiencing the sensation of touch. The APT nerve transmits information via electrical contact. Electrical contact refers to the flow of electrical current in an object. During the testing of the APT nerve, response resistance was recorded in order to demonstrate the response elicited by the artificial nerve, analogous to the response of an actual biological nerve. Response resistance is the measure of resistance to flow of electrical current, typically measured in ohms. The use of testing response resistance allows researchers to measure current flow in response to mechanical stimulation. The research aimed to address these two major questions. How can an artificial nerve be created as simply as possible that is as similar in function to a human biological nerve as possible? And how does the APT nerve detect mechanical stimulation, consume low or no energy without mechanical stimulation, transmit mechanosensitive signals, and recognize the location of mechanical stimulation? To answer these questions, the researchers first designed the nerve they would be testing. The APT nerve consisted of two graphite-based conductive films which were face-to-face taped together and served as the active layers. Overlaps or cracks were generated by bending the paper so that the resistance of the conductive film would decrease or increase correspondingly. Therefore, the overall resistance of the APT nerve would be relatively stable, and thus the APT nerve could operate in a bending state. To test the capabilities of the nerve, the researchers performed many short-term and interval mechanical stimulation tests and tested the response resistance of the nerve. In one experiment, they tested the relationship between response resistance and location of mechanical stimulation. They also tested how response resistance would change when they changed the thickness, width, and length of the nerve and how quickly it responded to the stimulation. They also applied stimulation at different locations of the nerve to see how the location affected response resistance. The researchers conducted over 10,000 tests in order to ensure the durability and robustness of the nerve. The researchers found that the relationship between response resistance and the location of the mechanical stimulation was nearly linear. As can be seen in Figure A, when the thickness of the spacer was increased, the response resistance slightly increased. In Figure B, when the width of the active layer was increased, response resistance slightly decreased. And in Figure C, it can be seen that the difference in length didn't have much of an effect on the response resistance, and a linear relationship between response resistance and location of stimulation was relatively maintained throughout each trial. Because of this, the researchers fixed the thickness of the spacer and the width of the active layer as 0.12 mm and 5 mm. The interval stimulation also revealed that a threshold of at least 5 kilopascals of stimulation had to be applied in order to allow the signal to propagate the current. It can be seen here that during different time intervals, the APT nerve showed a fast, stable response resistance in response to stimulation. When stimulation was applied at 3 centimeters on the APT nerve, the response resistance was lower, while at 6 centimeters, the response resistance was higher because of the linear relationship between response resistance and location. In the last graph on the left, it can be seen that when 10 kilopascals of stimulation was applied, the nerve responded in a little less than 21 milliseconds. When the stimulation was removed, the mechanosensitive signal disappeared very quickly as well. Similar to the recognition of the signal, it disappeared in a little less than 21 milliseconds. These tests show how the artificial perception and transmission nerve has very similar characteristics as those of the biological nerve. We can see the ways that the APT nerve incorporates the important qualities of a biological nerve that were previously mentioned. The nerve can detect mechanical stimulation through electric contact. When a stimulation is applied, the two layers make contact, causing electrical current to flow and therefore mechanosensitive information. We know that the nerve is detecting a signal because of the tests of response resistance. The test of the speed of nerve response and dissipation shows how the APT nerve consumes very low energy without the presence of stimulation, which was an issue faced by other artificial neural systems. It only consumes energy during the stimulation, similar to the way a biological synapse consumes energy. The APT nerve can also transmit mechanosensitive information. The researchers show this by showing that, to propagate the signal, the nerve must receive at least 5 kilopascals of pressure from stimulation, which is similar to the way a neuron has a threshold of depolarization in order to cause an action potential. The current will flow when 5 kilopascals are applied and mechanosensitive information is transmitted via the electrical current. The APT nerve was also shown to be highly robust when transmitting information by the way the response resistance was stable during different time intervals of stimulation at the same location. If the response resistance varied greatly, this means the nerve would be highly affected by interference when transmitting information. The quick changes in response resistance of the nerve was similar to the excitation and inhibition of a biological synapse, which was also activated by the external stimulation and then returned to the original state after removing the stimulation. Finally, it was shown that the nerve could recognize the location of the mechanical stimulation by the linear relationship between response resistance and location of mechanical stimulation. As the location moved away from the electrodes, the response resistance would increase. By analyzing the magnitude of the response resistance, the APT nerve could recognize the location of the mechanical stimulation. Since the response resistance was correlated with the location of mechanical stimulation, the APT nerve could also mimic the spatiotemporally dynamic logic of a neural network. These similarities show how the nerve can be a very useful alternative to typical artificial neural systems, and it can also provide advancements in artificial intelligence like user identification, feedback control of prostheses, and the intelligent action of a robot. There are many future possibilities for advancements of the baseline APT nerve described. The researchers indicate that the combination of different functional materials for fabricating devices could even enable the APT nerve to feel sensations such as temperature and humidity. They also discuss creating it with other organic materials to allow it to be more stretchable. The robustness and durability of over 10,000 cyclical tests enabled the APT nerve to operate for highly stable and reliable mechanosensitive interactions and to avoid frequent maintenance. The separate electrical double-layer structure made sure that the APT nerve featured no energy consumption when there was no mechanical stimulation, which could achieve long-term work. In these ways, it avoids the issues presented by the typical artificial neural systems and functions like a biological nerve. High flexibility allowed integrating the APT nerve with the human body to be as comfortable as needed. The rapid response time and high variability in shape gave the nerve the potential to help disabled people regain their ability to feel the sensation of touch. I believe the researchers did an excellent job with this research and would like to applaud them for finding such an innovative alternative to typical artificial neural systems. My only critique of this experiment is that I would like to have seen testing in live subjects in order to understand exactly how their nerve would work when incorporated with the nervous system. I feel that this would have provided readers with a greater understanding of the real-life applications of this nerve in artificial intelligence and neuroprosthetics, which were the applications of focus of their research. Other than that, I believe the experiments conducted to demonstrate the way the APT nerve functioned did a great job in showing how it is analogous to a biological nerve.